Kia ora, this is your introduction to the Biology 213 Plant Symbiosis Lab, which you need to watch before coming to lab and make sure you understand the procedures and what you're going to do. For the lab, our objectives in terms of a research objective are to quantify the arbuscular mycorrhizal colonization and the bacterial rhizobial nodulation in plants, and to assess the presence of potential pathogens in our plant roots. And those research objectives are useful in understanding whether plant symbioses might limit plant restoration efforts going on in the red zone, and also just understanding the biological responses of the red zone to the changing environment. In terms of learning objectives, you'll also be learning skills in histology, building your skills in microscopy, and increasing your understanding of two of the major plant microbe interactions that drive ecosystem function. We will be working with the pea plants that you planted in soil from your sites out on the red zone. There should be 2 plants, a house plant and a garden plant, for each pair of 2 students, and you are going to be working in teams of 2. If you don't have 2 plants for whatever reason, say if your plants died or if you didn't do it properly during the first lab, see me. We do have some control plants that need to be measured, and you can do that instead. You're going to start, before you do anything else, by measuring the plant height and assessing your plant vigor, and there is a score sheet in your lab manual showing you how to do that. Then you're going to start removing the dirt. We need to contain the dirt. We don't want this getting everywhere, so please do this over a tub. We're going to do it dry first. Don't tug on your roots, just sort of gently crush the roots, getting the soil to break apart, and catch all of the dirt that comes off into a dish where we can then take it back outside. You're going to get a lot of the dirt off by doing that. So you're down to just the root level. There'll be some dirt that's still adhering. At that point, you can move to a clean bucket of water and put the root system into there to get most of the remaining dirt off of the root system. Please do not pour that water down the sink. We will collect it in buckets, again, so that we don't clog the drains. Even after that, there'll be a little bit of dirt that stays on. You can put the root system into a petri dish and use forceps to clean off those remaining chunks. At this point, you're going to want to look at your instructions and go through the nodulation assessment, looking at whether the nodules are found near the crown of the plant or more laterally out inside the root system, or both, the color and the abundance of nodules. You also will need to set aside some nodules for later assessment. You'll just need about 4 or 5 of these set aside that you're going to measure at the end of the lab. Once you do that, you're going to take some of your roots and you're going to put them into a histology cassette. When you do this, you want to take the smaller roots because that's where the mycorrhizae are really going to be found, the first 3 orders of roots from the tip backwards. Don't worry so much about getting the really big ones because they're going to make your slide too thick. They don't clear very well, and they also don't have a lot going on biologically. Don't chop up your roots. Try to keep them connected. Those fans of connected roots will help keep the roots inside your cassette, whereas if you do chop them up, we find they tend to wash out of the cassette. You lose most of your sample. These cassettes come with a lid attached to one side. You actually snap that lid off, sort of turn it around, and then it's got 2 little tabs that you can insert into the openings that are there for it and click it together. Once you get your cassette full, label it with the house or garden name, number. Make sure you label it with a pencil because pens will come off in the KOH stage, so it has to just be a normal pencil. And then you're going to add this little wire hook. With the wire hook, it's like a tea bag, and a tea bag has a string and a label on it. We're doing that with wire. That's so that we can hook it over the edge of the beaker, and so that you can see the label on the outside of that hook, so that when you need to pull the sample back out of the beaker, you can find it. Go through all of those steps for both of your plants. Get both samples ready. Once you have both samples ready, you're going to take them over to the KOH bath, and you're going to put them in just boiling KOH, and you're going to leave them there for 25 minutes. Ideally, you want the temperature to be at a sort of gentle simmer while they're in there, so that the KOH is sort of slowly moving around, maybe a few bubbles coming, a bit like that. Gloves and eye protection are essential at this stage. Boiling KOH is highly caustic, can cause permanent damage to your eyes, and isn't pleasant on your skin either. After the KOH stage, you're going to let the KOH drip back off into the beaker, and then you're going to transfer your cassette into a second beaker where you've got a bit of simmering vinegar and ink solution. This is going to stain the fungal tissues. And you want to do this for 3 to about 5 minutes. 3 minutes should be plenty. Whenever you are transferring these between the beakers or taking it back to your bench, we've given you these little blue bowls. That's so you can avoid dripping the hot KOH and the ink solution on the floor or any surfaces. So please use those to contain spills. When you come back to your bench, you can open the cassette, and then you can put the roots into a petri dish and add a little bit of water. Degassed water is ideal for this, and sort of gently tease your roots out. Now your roots are going to be really soft at this stage because they've been cleared in the KOH, and so you need to be very, very gentle. You're then going to have a hot plate on your bench with a glass plate petri dish on it. You're going to put a glass slide on the hot plate in that petri dish, and you're going to use this mounting media called Kaiser's jelly. Kaiser's jelly is liquid when warm, and then as soon as it cools to room temperature, it will solidify. Take a few drops with the eyedropper and spread them over about 2/3 to 3/4 of the slide length. And the whole time, make sure this is warm. Not too hot. It shouldn't be so hot you can't touch it, but it should be nice and warm so that the Kaiser jelly remains gelatinous or jelly-like, liquid. You're then going to use the tweezers to very gently transfer the roots from your petri dish onto the slide. This is a really tricky step. It's going to take a little care to do it properly. What you want to achieve is so that you've got your roots spread out on the slide. This is just adequate. Spread out, it's going along the long direction of the slide in a whole bunch of rows. Get on a good number of roots onto your slide. You don't want any roots to overlap each other. They have to all be visible separately. Then you're going to take one of our large cover slips. These are much larger than a normal cover slip, and add it to your slide. And with a gloved finger, gently compress it over the warm dish. You can then take it off of the hot plate and let it cool on your bench. Now, sometimes some of that jelly is going to squeeze out on the edges if you put a little bit too much on. Don't try to remove it right away. Let it get fully cool, and then if it's in your way, you can either slice it off with a razor blade or sort of peel it back. If you do that while it's still hot, you're going to just smear it on your slide and you'll have to start over. Now you can go to the microscope, onto the compound scope, and you can look for different root structures. Take a little bit of time to look at your sample, get familiar with what you're seeing, and we'll talk about those in a minute. And then your goal is going to be to record the abundance of different structures in the root on the data sheet. This is most easily done if one person is at the microscope and they're telling their observations to the second person, and the second person is writing it down. In order to do that, once you've become familiar with the root system, you sort of know what you're looking at, you can use the stage controls to move the slide across your field of view, and you're going to move it across the short dimension. Every time that you hit a root, you're going to record a root in the first column, so you can put a tick mark there, and then you're going to decide what structures are present, whether you have hyphae, vesicles, arbuscules, other fungi, or oomycetes. If you hit a root and you just cannot tell what it is, the root's been degraded or it's really, really dark and you can't see any structures, you would record that you have a root and that it's unclear. But only record unclear if you don't record any other structures. Once you complete one pass, you can just go forward and backward across the root, recording what you're seeing each time you go across until you've got about 50 intersections of root with your field of view. Just to illustrate that, if this is what you were seeing under the microscope on the right, there's a transect going across there, and I can see that it's crossing a root, so I'd record one under the root. I can also see that there's some hyphae there, particularly at the bottom of where it's crossing. And then it's crossing a sort of fuzzy structure that's filling the plant cell, and that's actually an arbuscule. So I would record for this spot a tick for root, a tick for hyphae, and a tick for arbuscule. Then I might come back in another spot, and there I see that sort of rounded structure, that's a vesicle, and I also see some more hyphae, so I would record a tick for root, a tick for hyphae, and a tick for vesicle. What are these structures? Ideally, they'd look a bit like this. There's a few different structures you can see here. The hyphae of arbuscular mycorrhizal fungi are sort of knobbly. They should be stained a dark color. You'll notice in this top picture that there's some things labeled RH. Those are root hairs. They're actually kind of a sign that normally that a plant doesn't have mycorrhizae, because the mycorrhizae tend to suppress the root hairs. But you notice they're not stained, and they're quite a bit larger than the hyphae. The hyphae are going between the plant cells, and then inside the plant cells, you see these sort of fuzzy blue structures. They don't look like you might expect from a textbook picture where scientists are giving you the very best picture they've ever taken in their whole career. They tend to look like a fuzzy blob inside a cell, but the arbuscules sort of fill the plant cell. They don't have a defined edge to them. And then you have these a little bit easier to see rounded structures that have a nice defined edge. Those are vesicles, and that's a spot where the fungus is storing lipids as an energy source. Both vesicles and arbuscules will always be associated with hyphae. If you see something round just by itself with no associated hyphae, it's almost certainly not a vesicle. What it probably is some other fungal structure. Now, to be honest, we're not exactly sure what all of these things are, but I think that a lot of them are some form of oomycete or other protist. We're going to record them either as oomycete, if they're sort of these round structures like these, or if you see some brown hyphae with no arbuscules, we might record that as other fungi. So hopefully that will work out for you. You can also look online for some additional photographs to get a sense of what to look for. If things aren't working, there are a few things you can check. Sometimes if your hot plate was too cool when you were making the slide will be really thick. And you just need to put it back on your hot plate, let it warm up, that will liquefy the media, and then press down gently with a gloved finger to get it a little bit thinner. If that doesn't help, check the microscope settings. Is your condenser set for transmitted light, or have you accidentally put it into phase contrast? You should not be using the phase ring, so it should be slid all to one side, so you're just doing normal transmitted light. Lastly, check that your light is properly aligned, that you've colored your light, and if you've forgotten how to do that, we've covered it in previous labs. With that, you should be able to get your data for mycorrhizal infection. We also want you to look at the nodules and assess the presence, size, and shape of rhizobia. To do that, you're going to place a couple nodules on a slide. You're then going to take a razor blade and sort of mush them up, cut them up, and smash them into a droplet of water. Use sterile water for this. And then you're going to transfer I often find the end of a toothpick works really well, just a little bit of that liquid to a new slide. That's the step people often forget to do, is that transferring of liquid. And then you put a cover slip on it using one of our normal square cover slips, not one of those expensive big ones. With that slide, then, you can look at it under the microscope. Here you're going to want to use phase contrast, and you're going to want to go up to oil immersion, so 100x. We've asked you to, just remember that you'll need to slide that phase ring over to phase 3 and also switch the condenser over to the phase setting. You can use the camera to measure what you're seeing in the microscope. Just remember you need to go up and set the scale first, as I'm showing you in the top of this image, and then you can measure using the line tool. You want to record the shape and the size of the bacterial cells and any other observations you have. Those are the key tasks for this lab. Just a couple reminders. Safety first. We really need to be careful in this lab. We've got the hazard of the KOH, so you want to make sure you're using eye protection and also gloves when you're dealing with that KOH. We're also using razor blades. We use single-edged razor blades because they tend to result in fewer accidents. But if you haven't used these before, you'll notice there's a metal side that you can see. That's the handle. It is not sharp. And then there's a part wrapped in sort of a brown cardboard. That's where the sharp edge is. You want to open that razor blade by peeling the cardboard off, starting at the handle side of the cardboard. That way you're protecting your fingers from that sharp edge. You also have sharp wires that you also need to watch out for. When you're done with your razor blade, make sure you put it somewhere safe. Don't leave it lying around. And when you're done at the end of the day, it needs to go into the sharp container. Your used slides can go into the used slide container, but the wires we reuse, so please just put those back within your equipment tray at the end of the day. Hopefully that gives you a quick introduction, and I'll look forward to seeing you in lab.